Hey everyone, I am recording this video to walk us through the Decoding the Flu case study. Now I've uploaded the PowerPoint and I have uploaded this flu decoding answer sheet here. The first 2 pages are just some facts about the flu that you can peruse at your leisure. Then the last 2 pages are a student handout for codon problems. So if you see here, there's practice problems 1, 2, and 3 on here. I suggest printing out at least these last 2 pages and to make it easier to write on, because you'll have to mark where mutations occurred and decode some of the strands. So do that. If you want to have the print copy, if you're okay having just the digital, that's up to you. Now you can also view this anytime you want. I'm going to make sure everything is getting shared properly. So, just to double check, share my screen here. All right, so Jason was worried. He had landed a summer internship on a CDC research trip to rural Mexico. However, what had appeared to be a wonderful opportunity didn't seem so great when the team contracted one of the flu viruses they had been studying. So far, he was the only person other than the team leader, Dr. Phillips, who was not sick. Here comes Dr. Phillips. Normally, I would give this to a senior staffer, but they're all sick. We think there might be a problem with the flu virus the team has caught. For starters, the flu virus is atypical. The symptoms are worse than usual. None of the team's vaccinations give them any protection. This may be a new strain of flu. I need you to find out. The hemagglutinin HA protein helps the flu virus infect cells, and the structure of this protein can vary in different virus strains. I want you to compare the HA gene for the viruses the team was examining with a typical flu virus. I will give you the nucleotide sequence for a typical HA gene. You can start by finding the coding region for the gene. Because we don't have power right now, you'll have to do this the old-fashioned way with pencil and paper. So what is a gene? So genes are short stretches of DNA on the chromosome. We have our regulatory region and a coding region, shown in red and green. And the information in genes is used to make proteins going through the 2 stages of transcription and translation. So, question 1. If the DNA molecule above were transcribed, which of the following RNAs might be produced? I'm going to give you all a chance to look at this. I'm going to try to move my stuff there. So I want you to think about which direction a template strand needs to get read to be able to make the new strand in 5' to 3' direction. So with that in mind, if we started from this strand, we would be adding on, the new strand up here would be 5' end, T, A, G, well actually it wouldn't be T, it'd be U, sorry. So that already should tell you then some of the strands are going to be different. RNA is going to carry uracil, not thymine. That means we can rule out A and B already. Got your answers? All right. So looking at this, if we transcribed the first strand, the top strand, into RNA, we have our 5' end over here, to 3'. Now the bottom strand would really be going from 5' end, adding to its growing 3', it would go this direction. But then it could get flipped around on the next part. So we have our answers there. I believe that means it should have been both C and D. AUCG, AUCG, yes, so answer is F, both C and D. So now translation is going to turn our DNA RNA into protein. So if a combination of 2 nucleotides corresponded to an amino acid, how many different amino acids could DNA code for? So what we're looking at here is, let's try to see if I can do this as a whiteboard pretending this is my board, we'd have two strands, or two nucleotides, 1, 2. Now, we have 4 options, A, T, C, G, or I guess A, U, C, G for the first one. And then we would have 4 options for the next nucleotide. That means all in all, we would have 16 options. 16 different combinations. That's saying that AA is one, AU is the next, AC, AG. And then we'd have started them all again with the U's for the first letter. All in all, we would get 16 different amino acids. So, with our 16 amino acids, if we only had one letter code, A, G, C, U, we would only get 4 choices for amino acids. With our 2-letter code, using the math that I just showed you, we would have 16 options. Now we use a triplet code. That means we have 64 different options. Now we don't have 64 different amino acids, but we have 64 options for them. Messenger RNA carries the nucleotide code for the proteins to be made. Transfer RNA, tRNA molecules interpret the nucleotide code. Now, each tRNA carries an amino acid and an anticodon complementary to the codon in the mRNA. So down here, we had our mRNA in the 5' to 3' direction, and it's just showing randomly 3 of the letters. Our tRNA would have an amino acid here at the top with anticodon down here at the bottom. It's part of loop 2. Remember, if this 5' is this way, because they have to be opposite directions, 5' to 3', so this one has to go 5' to 3' that way. Part of an mRNA molecule is shown above. If this fragment came from the beginning of the mRNA, for which of the amino acid sequences below would it most likely code? So now we can go back and look at one of our pages here and be able to see our letters. Now, if we started from the beginning, we have to look at which end do we go from, 5' or 3'? We always read it 5' to 3', just like the mRNA shown here, 5' to 3'. So we could have the choice of starting here with CAC. So first base C, second base A, third base C would give us a histidine. But I think some of y'all might know the problem with that. I ask you to look through this and hope that some of y'all would be able to raise your hand. Raise your hand if you can read this. Not all of an mRNA molecule codes for a protein. We need punctuation to identify where the coding region begins and ends. So we need to find our start and our stop codons. So AUG is our start codon, and we have 3 options for stop codons. So going back, we need to find the AUG. AUG is right here. That's going to give us our methionine to start things off. C is the only choice that has methionine in it, or starts with methionine. Now, looking at this, this is saying that you weren't looking for your original open reading frame that starts with AUG here. And we could have 3 possibilities. You could go directly CAC, you could skip the first one and go ACG, or you could skip the first 2 and get from CGG. That would be histidine, glycine, arginine stop. And so these were some of the different possibilities that were listed out. Now we know ours needs to start with methionine. Without computers, all I've been able to find so far is the partial sequence I believe is at the beginning of the HA gene. I also have the start of the protein sequence. So she gives us a DNA sequence. Remember, our DNA sequence is double-stranded, and we have the start of the amino acid sequence here. We see it starts with methionine, so that should tell you what you want to be starting with. See if you can find the coding region. Now, I do believe if we bounce back over here, onto the flu fact sheet, you have this information right here. This is the amino acid sequence that is given in the PowerPoint, and this is our first stretch of DNA. I want you to find the beginning of this coding sequence. In fact, find out where it begins, which direction it's going, because remember, it can be on either strand. Now, you don't hit the end of the strand and wrap around. You can imagine that this is ... It'd be part of a much larger chromosome in both directions. But this is just for you to practice. I'm going to go ahead and stop the video here at this part to give you a chance to work on it and give me a chance to not make this video ginormous.